All right, guys, this one's probably going to be one of the quicker ones for the viruses, even though there was a lot of information to cover with viruses. Yeah, as far as the review goes, I feel like it's a pretty straightforward one. There's a lot of terms involved in this one. So remember that viruses are obligate intracellular parasites. A virion is a single virus particle that is like able to be infectious. It's fully infectious. It's fully formed. It's ready to go. A genome, that's a term that's referring to the entire genetic makeup of an organism, including any extracellular DNA. For viruses, these can be single or double-stranded RNA or DNA, right? So we learned about all the different kinds of genomes that viruses can have, and it is completely different from what we are used to seeing with cells. So that's pretty crazy, and they require completely on the host cell for them to be able to replicate and everything like that. So that's really what obligate intracellular parasite means. They depend entirely on that host cell for everything. Obligate means they have to, intracellular inside of the cell. Parasite means it's causing some sort of detriment to the host. Then we have the capsid. Capsid, so we know that viruses have to have two things. They have to have a coating, and they have to have their nucleic acid, their genome, right? They have to have those two things at a bare minimum. So their coating has to consist of at least the capsid, that is like the protein shell that surrounds their genome. And when you have just a protein shell surrounding a genome, that is called a nucleocapsid. So that's what that is. Some viruses, so all viruses also have spikes. Those are glycoproteins, so sugar plus protein together. They work together as a key to enter the door that whatever they match up to on cells that they're targeting. And based on what door they can open with those spikes determines what tissue they invade and what host they can invade and everything like that. So that's all determined by their spikes. Some viruses also have an envelope, which is just they coat themselves with usually the envelope, the membrane of the host that they infected. So they bud out at the end after they make new viruses. They bud out and push through the membrane, and that membrane is usually coated in the spikes of that virus. And then they just pinch off and they can go and be viruses now. So that's where they get those envelopes. Not all viruses have envelopes. Cytopathic effects, these are just terms referring to changes in the changes in cells due to the effects of viral infection. So you either being coated by that spike protein on the outside of the cells, allowing them to form syncytia, which are merging of cells together, like becoming one. So you can have multiple nuclei in this big mass of what used to be several cells, usually due to the viral proteins causing the cells to fuse together like the viruses would into the host. And then, or like overproduction of stuff within the cell itself, causing damage within, or formation of inclusion bodies, which is like trying to, control the damage that's being done to the cell. All of those would be considered cytopathic effects. Bacteriophages, these guys are viruses that infect bacteria, and it looks like all bacteria ever have at least one bacteriophage that can infect them. These guys also always lyse their host cells. So you know how I was talking about how envelope viruses push their way out nice and easy through the membrane of their hosts cells, right? And bud out. Yeah, the bacteriophages don't do that. They just bust out and like bust out of the cell. So that's how they get out. But most of us have like the slow outbreak of the budding viruses. So that's the eukaryotes that have that. Bacteria, not so lucky. All right, lysogeny and lytic cycles. The lytic cycle is exactly the busting out version, right? So you have your actual virus that is coming along, infects the bacterium, and injects its DNA into the cytoplasm of the bacteria, because they don't infect the whole virus, doesn't go in. They just inject their genome in. So they inject their genome in, and if they go from genome to making protein parts of the virus using that genome, then assembling those together and then forming new virions that can just burst out of the cell. That's the lytic cycle. The lysogenic cycle, however, if we inject our genome into the bacterial host cell and then have our genome incorporate itself into the DNA of the host genome as a prophage, then that bacteriophage is considered to be a temperate phage, and that would be called a lysogenic cycle whenever you had that creation of the prophage. So that can stay that way for a long time, and that can even impart the ability of the bacteria to make like things like toxins, which is actually pretty medically significant. So that is lysogeny versus the lytic cycle. And there is anytime that the bacteria decides to go from lysogenic, incorporated within the genome, and switch back to the lytic cycle, where it's going to form viruses and burst out of the cell. That switch is called induction. Proviruses. So proviruses are basically prophages, but in eukaryotes. Okay, so viruses that have incorporated their own DNA into our DNA. There's quite a few viruses that do that, including things like chickenpox and herpes viruses in general do that. So that would be latent, that would lead to a latent viral infection, and those can come back out way later on in life as well. But yeah, so obviously it can come way later on in life, or you can have like a herpes outbreak, I don't know, due to stress or something like that. Not uncommon. Oncoviruses, these are viruses that cause cancer. Could lead to development of tumors. We know, most of us are aware of cervical cancer. That's a pretty common example of this, right? Not unheard of. And we actually have vaccines available for a lot of the more common strains that cause this. So I highly recommend that. And they even offer it to people who have already been sexually active now, because there's still strains out there that you may not have encountered. So we can still protect you from those ones. Remember that viruses are simple, non-cellular, so they're technically not alive, they're non-living. They lack organelles completely. They lack ribosomes. They don't have their own ribosomes anyways. Some viruses are capable of stealing ribosomes from the host that they can bring with them, but that's not their ribosome. They did not ever make it. So that's viruses. And then we also talked about the other non-cellular infectious things. And this would include prions, which are infectious proteinaceous particles, essentially misfolded proteins. It seems like we think that can cause spongiform encephalopathies, which are subacute encephalitis, essentially, but long time to develop, and I don't know, anything like that. So that's that for chapter 7. And I will see you guys for the next one. Bye.